This is podcast 30. Okay, we just talked about mitosis and in the cell cycle how there were different phases of the cell cycle. G1 is the first growth phase. If cells maintain most of their life in the first growth phase, that's called G0. S is the DNA is duplicated, like we talked about, how DNA polymerase and the origin and replication, and you get loops forming. That happens during the S phase. And there's a second growth phase, and then mitosis. There are checkpoints in the cell cycle that the cell makes, quote unquote, decisions about if it should enter the next phase. There are cell mechanisms as to check on these things. And if you think about this, this would be selected for in evolution. So for example, if a cell was spending most of its life in G0, there's a lot of things that could happen in its exposure to the environment over years of its life, for example. And one of those things could be that its DNA is damaged. And so you would want to check that before you duplicated your DNA, because you might be duplicating a mistake. Similarly, before you enter mitosis, you want to see how did the synthesis go? Was there a large mistake in that synthesis? And then during mitosis, there are certain things that have to happen, or mitosis isn't going to proceed correctly. And if you allow it to proceed, you can end up with a cell with an uneven number of chromosomes, for example. So, for example, entering the S phase, this can be influenced by, has the cell grown enough? Are there nutrients in the environment available to grow a new cell? So if I duplicate this new cell, will it have something to eat? Has the cell reached a certain size, and is it time to duplicate? And again, like I mentioned before, has there been significant DNA damage as the cell was resting, or not resting, but in growth, the first phase of growth? Same thing in G2. What's the size of the cell? Has there been DNA damage from the synthesis phase? And has DNA been replicated? Did S happen? In the metaphase anaphase transition, did you split those chromosomes correctly? Did half of the chromosomes go to each side of the cell? These are the sorts of things that there are cell mechanisms in which to check. And when these mechanisms fail, a lot of them can be carcinogenic, can be resulting in cancer, because part of controlled cell growth is going through these checkpoints and the cell checking off its list of everything is okay. If things are not okay, that is not a cell you want to duplicate. And if you go past the checkpoints without checking those things, one of the reasons you could be going past is because the checkpoint itself has a mutation in it and can lead to uncontrolled cell growth, or cell growth without going through these checkpoints. In the research where these checkpoints systems were discovered, they started with some early experiments where they take cells that are in different parts of the cell cycle and they would fuse their membranes together. So you take a cell that was actively in the S phase, you fuse it with a cell that is in the first growth phase, and what would happen is the growth phase nucleus cell would start to go through S as a result of that. Or you could take a cell that was in mitosis, fuse it with a cell that was in the first growth phase, and the cell that was in the first growth phase would start to go through mitosis. This was known as maturation, and this activity was called a maturation promoting factor, or MPF. Biologists often call things a factor when they can't they don't know what it is, but just know it as a phenomenon. Later on, we found out that this factor was in fact a kinase, and the kinase was made up of two different parts. So if they check the ability of this fusion to induce maturation, it wasn't constant across the cell cycle. So cells that were entering mitosis had the ability to induce other cells to enter mitosis. But if you fuse one G1 cell with another G1 cell, not much happened, and so on over time. When after cyclin was discovered, which is a protein that's important to this cycle, it was found that the concentration of this protein also went up and down in concert with this ability to promote maturation promoting factor. And then after the first cyclins were discovered, many more cyclins were discovered that were similar in structure, but different activity at different parts of the cell cycle. So here are all kinds of different cyclins: cyclin A, cyclin B, E, D1, D2. They all wax and wane over different parts of the cell cycle. So D2, for example, starts to build up in concentration during G1. It peaks right at the G1S transition and then degrades over time. So this cell cycle progression is governed by synthesis and specific destructions of cyclins. So it's not only important that the cyclin get made, it's also important that it get destroyed in order for the cell to make it through these different checkpoints. The cyclins play a role in a kinase, which is a phosphorylating enzyme, and these kinases are called cyclin-dependent kinases, or CDKs. So these are kinases whose phosphorylating ability depends on the presence of cyclin. When cyclin is bound to the kinase, it can phosphorylate things, and after cyclin is destroyed, such as in the middle of mitosis for cyclin A, then that kinase goes away, or that kinase activity goes away. Okay, so again, for this particular cyclin, it builds up in concentration, then crashes, and when it crashes, the ability of maturation promoting factor, which is just a kinase, that crashes with it. So as cyclins build up, you get the ability to promote maturation in cells by fusing two types of cells together. And then as a cyclin is destroyed, the ability to have kinase activity is also destroyed. Okay, here is a model, and you can see lots of these different things in different parts, in different books. But there's the kinase part of the protein, the CDK, and then there's the cyclin part. And the cyclin has to be bound to the kinase for the kinase to be active, for it to phosphorylate things. If the cyclin is degraded, it's destroyed, and we'll talk about how that happens, then the cyclin-dependent kinase is no longer able to phosphorylate things. It is cyclin-dependent. So if the cyclin is gone, it can no longer form a kinase. So at the G2M transition, leaving the second growth phase and entering mitosis, the cyclin-dependent kinase will cause nuclear envelope breakdown. It'll cause the chromosomes to condense. It'll cause the stimulation of polymerization of the mitotic spindle, and it will cause certain proteins to be degraded. Okay, these are all things that are known to be associated with the G2M transition, and they are as a result of this kinase. And there's a specific one. We're just going to say a a CDK in general, that is going to phosphorylate certain protein substrates in the cell, and that's going to cause these things 1 through 4 to happen. We'll go over one example of how that would happen. Okay. There's another protein system here called the anaphase promoting complex that is involved in promoting anaphase, just like it says, moving a cell through anaphase, and we'll talk about specifically how that works. So here's an H5P question for you. Okay, here's an example of how kinase activity can move a cell through the cell cycle. So one of the things that happens in prophase is the nuclear envelope degrades. And the nucleus has a shape, and that shape is maintained by a mesh network of these protein filaments called lamins. There's different kinds of lamins, A types and B types, and they will form on the outside shape of the nucleus and contain the chromatin material inside the nucleus. These filaments intertwine with each other, and they depend on phosphorylation or dephosphorylation. So when these individual lamin filaments are phosphorylated, the network that they form to form this mesh that keeps the nucleus together falls apart. And it doesn't destroy the protein, it's just the proteins don't interact with each other to form this mesh when they're phosphorylated. And that would cause the nuclear envelope to dissolve or disassociate is a better word. And then that's a characteristic of prophase. Later on in telophase and cytokinesis, when you're reforming a nuclear envelope in the nuclei that form in each daughter cell, you have a dephosphorylating enzyme, a phosphatase, that is going to remove the phosphates that were put on these individuals, allowing individual monomer units of lamins, and then allow them to repolymerize and reform a nuclear envelope. And here's another example of how phosphorylating something makes a difference in terms of moving a cell through the cell cycle. So the anaphase promoting complex is this complex that adds a tagging enzyme, or not enzyme, sorry, a tagging protein onto other proteins that will cause that protein to be degraded. So the tag that gets put onto these proteins is called ubiquitin, and the more ubiquitin tags you get put onto you, the more likely it is you'll be degraded, and we'll talk about that in the next slide. So the anaphase promoting complex, this thing here, targets securin. Securin is a protein bound to an enzyme called separase. And by targeting, I mean it puts ubiquitin molecules on securin. Because ubiquitin is tagged onto securin, that means that securin is going to be degraded, turned into amino acids instead of a whole protein. When that's degraded, that releases this enzyme separase. Separase then can attack the connection that holds sister chromatids together in chromosomes that have gone through the S phase. So S phase now have 2 chromatids per chromosome. They line up during metaphase. Separase is released as a result of securin being degraded. Separase breaks apart the protein cohesin that's going to hold these sister chromatids together, and now you have a chromosome with one chromatid going to one pole, and a chromosome with again with one chromatid going to the other pole. So what was one chromosome becomes two chromosomes as a result of the action of separase. Okay. Another thing that anaphase promoting complex does is it adds ubiquitin onto another protein, and that other protein is cyclin. And now that cyclin stops its gets degraded because this cyclin dependent kinase is dependent on cyclin, and cyclin is now gone. That cyclin dependent kinase turns off, and that can this reduction in cyclin-dependent kinase activities leads to cytokinesis, chromosome decondensation, turning back into diffuse chromatin, and nuclear envelope reassembly. Okay? So the cyclins and the securins are degraded by being tagged with ubiquitin, and then they're directed to a proteasome. So what does that mean? This is not from your book, but from this website. Ubiquitin is a small, I think it's 76 amino acids in that range, protein that is tagged onto other proteins for their destruction. So there's a 3 enzyme system that does that. E1 takes ubiquitin as a protein and attaches it to itself. E2 takes it off of E1 and puts it on E2, and then E3 takes the ubiquitin protein and puts it on to whatever is going to be tagged. So that's shown in this red squiggle as the target protein. In the previous slide, that target would be either securin or cyclin. That can happen multiple times, so you can get multiple ubiquitins on a target. That's called polyubiquitinated. That's a real mouthful. There is a large enzymatic complex in cells called the proteasome. It's big enough that you can see it with an electron microscope. And that proteasome has ubiquitin binding sites. And when it recognizes proteins that are bound with ubiquitin, it takes the ubiquitin off of those proteins, and then they're used to tag new proteins, and then it shreds that protein into small 3-4 amino acid oligopeptides, which are eventually degraded into amino acids and can be reused as proteins. This is it's not the only tagging pathway, but it's a very common tagging pathway for protein recycling, and these are for proteins that are made and used within the cell, not for proteins that are imported by endocytosis from outside the cell. If they came from outside the cell, then they would be degraded in a lysosome. But inside the cell, if they're just old or they're part of some signaling system like we're talking about as part of the cell cycle, they're very commonly tagged with ubiquitin and then degraded in a proteasome. Like I said, ubiquitin is not the only system. There's another system called SUMO, but it's a very common system, and ubiquitin gets its name is because it's ubiquitous. It's found everywhere. Okay, so how could I ask you about these things that we've talked about so far? One possible extended response question that you could practice is that you could think about each of these events like the nuclear DNA in a cell doubles and and ask yourself, does that happen during G1? Yes or no? Does that happen during the S phase? Yes or no? Does it happen during G2? Yes or no? Does it happen during M? Yes or no? M being mitosis. And just do that for everything on this list, and that would give you a good sense of if you understand when things are happening in different parts of the cell cycle.